Good afternoon, everybody. Thank you for being here now after lunch. Siesta time. Well, I'm co-chairing this session with Ron Dagan. I don't know whether you know him. And I am Javier Díaz Domingo from Valencia in Spain. And it will be a pleasure to share with you this parallel symposium on new vaccines in town. So the first presentation that we are having will be given by Mary Hamel. It will be about malaria. She works in the WHO, and therefore she cannot travel because of the new policies, but she is online. Good afternoon, Mary. The floor is yours.
Good afternoon. Thank you. I've just lost my screen somehow. There we go.
Do you hear us?
We can hear you.
Yeah, good afternoon. Thank you. Let me just, I just lost my screen share, so I'm just going to reshare right now. All right. Are you able to see my screen?
Not yet. It was on before.
Well, let me thank you in the meantime. I am Mary Hamel from WHO, as was mentioned, and I am the team lead for malaria vaccines. And I am sorry I can't be there with you. Sure. It is.
Are your slides with you or they are with us?
They're with me. Do you see it now? I'm not sure why it's not shared.
No.
Boy.
We almost saw it.
We saw the first one and then disappeared.
Do you see this one?
Okay.
Here it is.
Okay.
Right. Thank you. Sorry about that. So I just want to say that I have no conflicts of interest to declare. And I'm going to talk about the malaria vaccine as a new vaccine in town. Here's my outline. I'll begin with a background on malaria and interventions. I'll speak a bit about the WHO coordinated malaria vaccine implementation program that ran from 2017 to 2023. And I'll give a brief update on the status of malaria vaccine rollout across Africa. So despite tremendous progress made since 2000 in the fight against malaria, with the increase in global commitment, including funding, the introduction of highly effective treatment, the scale up of insecticide-treated nets, malaria still remains a major cause of child morbidity and mortality, and particularly in Africa. This figure from the World Malaria Report shows the estimated malaria deaths globally in gold and in the WHO African region in blue. And you can really see that malaria is a huge burden in Africa. In 2023, it's estimated that there were 263 million cases of malaria and almost 600,000 deaths. And tragically, there were over 432,000 pediatric deaths in Africa from malaria. In endemic areas, malaria is often the primary cause of pediatric hospitalizations. In many hospitals, it makes up 60% and more than hospital admissions, and a major cause of childhood death. Malaria has a major impact on economies of countries, on communities, and on families. And you can see, if you look at this figure, that from about 2015, progress has really stalled. There's several reasons for this. There's increasing population, of course, so increase at risk of malaria. There's also emerged insecticide resistance, and insecticide-treated bed nets are the cornerstone of malaria control. We're now even seeing some drug resistance emerge. And then there's a stalling in the ability of more children to access preventive care. So we are looking forward with the introduction and scale up of malaria vaccines to seeing this trend turn downward again. We have a number of interventions to prevent malaria for children who live in endemic areas. All of them are partially effective, and none of them are used 100% of the time. So on the pathway from being non-infected with malaria to being inoculated with by the malaria parasite, by the Anopheles mosquito and becoming infected, to developing disease and progressing through severe disease and death, we have a number of points where we can intervene. To prevent infection, we have vector controls, most prominently insecticide-treated nets, and now we have malaria vaccines. To stop infection or cure it quickly, we have chemoprevention, which is the intermittent dosing of a treatment dose of medication given either in particular seasons or throughout the year, or for children who've been admitted with severe malaria or severe anemia at discharge. And then when a child does progress and develops disease, when prevention fails, we have rapid diagnostic tests and highly effective treatment that can be used in the community or in the health facilities. And if disease progresses, we have referral and hospital-based management. But one of the big obstacles is that the time from onset of fever to death is very short. In a couple studies, it's been shown to be 24 to 48 hours after the onset of fever. And that's far too rapid in many cases for parents to recognize that the child is severely ill or is going to become severely ill and to seek the care that's needed. So prevention is really key. And that brings me to the RTSS vaccine and the malaria vaccine implementation program. So in 2021, WHO recommended the first malaria vaccine, RTSS, and updated that recommendation in 2023 with the recommendation of the second malaria vaccine, R21. The recommendation is for the use of malaria vaccines for the prevention of P. falciparum malaria, that's the most severe form of malaria parasite, that for children living in malaria endemic areas, prioritizing areas of moderate and high transmission because that's where we'll have the highest impact. And you can see on this map showing areas of moderate and high transmission that much of Africa has these areas. And then there are a few pockets outside of Africa where there's moderate and moderate transmission. The vaccine is recommended as a 4-dose schedule given to children from around 5 months of age. Primarily in areas of seasonal transmission, a 5-dose schedule can be used to optimize impact, and the vaccine should be considered always in the context of a national malaria control plans. When this recommendation was made, our Director General of WHO, Dr. Tedros, called this a gift to the world, and the media really exclaimed that this was a historic moment, which really acknowledged the over 60 years of development of the vaccine, the many researchers that were involved in it, the many participants in clinical trials, and the hope that a vaccine brings for such a difficult disease that remains despite tremendous effort. This recommendation was largely informed by the pilot implementations. These were large pilot implementations conducted in Ghana, Kenya, and Malawi, where the vaccine was rolled out by the ministers of health of those 3 countries. And then there were independent evaluations. And the findings from the first 24 months of that evaluation were published in The Lancet last year. So I want to talk a little bit about this cluster randomized pilot implementation, which was to measure feasibility, safety, and impact of the vaccine. And the pilots ran from 2019 to 2023. So this shows the maps of Malawi, Ghana, and Kenya where the pilots were conducted, with the areas in green being the districts or sub-counties where the vaccine was first introduced at the beginning of the pilot, and the areas in red being the areas randomized to serve as comparators. Community-based mortality surveillance was set up in all of the pilot areas, and where you see the red dots, there was sentinel hospital surveillance. And this was to collect information on impact and safety. The countries selected their schedule that worked best for them in their each of the 3 countries. And this shows how the malaria vaccine was adopted into the routine child immunization schedule in each of the countries. So usually countries have a birth dose of vaccination, vaccines at 6, 10, and 14 weeks, measles at 9 months, and then some very important second year of life vaccines. The malaria vaccine for Malawi was given at 5, 6, and 7 months of age, and then a 4th dose at 22 months. And for Ghana and Kenya, they wanted to use some of the visits that when parents were already coming. So they gave the vaccine at 6, 7, and 9 months of age, and then the 4th dose at 2 years of age. Now, 2 years of age or 22 months of age is past the time when children are usually coming in for vaccines. And we did see that it was very difficult to get high coverage of the 4th dose. Ghana decided at the end of the pilot to move that 4th dose to 18 months of age, coinciding to the time when they give the measles and meningitis vaccine and an ITN, and immediately saw a very high bump up of the 4th dose to 85% uptake. So this is what we saw through the administrative data, looking at vaccine uptake in the 3 countries. This shows the percentage of children in the target population of the 3 countries who received pentavalent vaccine in purple as a comparator, because there tends to be high uptake of pentavalent vaccine. This is dose 3. And then the 1st dose of the malaria vaccine in blue, the 3rd dose in orange, and the 4th dose in green. And what you can see is pretty good and rather, I would say, rapid uptake for a new vaccine at a new visit of the 1st dose. Some drop off between the 1st dose and 3 dose, and then really a hard time reaching children with this 4th dose. This 4th dose was considered quite important. One of the objectives of the MVIP was to understand the importance of that 4th dose. This 62% was really reflecting what happened in Ghana. Before that, they were around 50% coverage before they changed that 4th dose to match their measles 2 vaccine time period. So these are the summary of the results from the 46 months of vaccine introduction in the 3 countries. During that time, over 2 million children were vaccinated, so quite a robust set of data, and over 6 million vaccine doses were administered. So first, the vaccine was confirmed to be safe. There had been some important safety signals in the phase 3 trial, and it was shown that there was no evidence of a causal association of those safety signals with the vaccine. So that was very important. And then secondly, during this period when about 70% on average of children had been vaccinated and about 40% had received the 4th dose, there was high impact. We were able to measure a 13% reduction in all-cause mortality that was attributable to the vaccine. And it's really quite marked, this reduction in all-cause mortality for a vaccine against a single disease. And I think that reflects the heavy contribution of malaria to child mortality in these endemic areas. There also was a 22% reduction in hospitalized severe malaria, and no reduction in the use of insecticide-treated bed nets, no change in care-seeking behavior, and no reduction in the uptake of other vaccines. We also saw that vaccine introduction was equitable and extended the reach of malaria preventive tools so that during the MVIP or towards the end, we were able to see that over 90% of children were either sleeping under an ITN or receive the vaccine, or what we would like to see, both. So I want to move to an innovative way that the vaccine is being delivered or was delivered in a clinical trial that optimized efficacy. Alassane Dicko and colleagues from Mali and Burkina Faso conducted this phase 3 trial looking at the seasonal delivery of the vaccine in areas of highly seasonal transmission, where malaria is really limited. Malaria illness is really limited to about 4 months of the year, with the remaining months being during the dry season. So there's intense malaria transmission over just a limited number of months. And the investigators aim to leverage the period of highest efficacy after vaccine is delivered and before waning of the vaccine efficacy begins to this period with high malaria transmission. So they gave the first 3 doses of the vaccine just before the high transmission area, just before the high transmission period, and then subsequent annual doses again before the high transmission period. And what they found was that this did result in high protective efficacy with a reduction in clinical malaria of 72% over the year after the first 3 doses were given, and continued high efficacy such that after 5 years or during 5 years, the efficacy was 58%. There was also high impact against severe outcomes, with protective efficacy against severe malaria admissions being 67%, and a reduction in all-cause mortality by 45%. So this is an important way in these areas to optimize vaccine impact. And WHO does recommend that malaria vaccine can be provided using these different approaches. So the age-based approach, as was used in the pilots, the seasonal approach, as I just mentioned, or maybe more practical than the seasonal approach is a hybrid approach, where the first 3 doses are given from 5 months of age, about a month apart, and then there are annual seasonal doses, and Mali has just launched the vaccine in this way. So if we look at data from different efficacy trials, we can see that highest impact can be achieved by using a mix of interventions, which is what WHO recommends that countries do. So if you have an area that has a certain burden of malaria and ITNs are introduced, malaria can be reduced by almost half. If you introduce ITNs and the malaria vaccine, or ITNs and seasonal malaria chemoprevention, malaria can be reduced by about 75%. And if you are able to introduce ITN, seasonal malaria chemoprevention, and vaccine in these areas of highly seasonal transmission, the malaria burden can be reduced by over 90%. So we would like to see countries have the opportunity to introduce multiple interventions according to what works in their country. So in October 2023, WHO recommended the second malaria vaccine, R21 Matrix M. The WHO was able to leverage a lot of the learnings from the MVIP to support the review of that vaccine. And what was found during 18 months follow-up is that the vaccine has a good safety profile. It has a good efficacy, reducing clinical malaria by 66% when given in an age-based 4-dose schedule, and by 73% when given seasonally in areas of highly seasonal transmission. Modeling estimates high public health impact with this vaccine, and the cost effectiveness is comparable to other malaria interventions, including ITNs and other childhood vaccines. And I should have said that both of these vaccines are similar in construct and target population, and both are pre-erythrocytic vaccines. So they aim to stop the malaria infection before symptoms begin. The high manufacturing capacity and lower vaccine costs of the R21 vaccine, which costs less than half of the cost of the RTSS vaccine, really is allowing more countries to scale up with this vaccine. And the countries are scaling up. Gavi reports unprecedented demand for malaria vaccine already, with the first non-pilot countries starting to scale up in 2024. 20 countries have introduced malaria vaccines, as seen on this map. Most have begun subnationally and are planning to scale up in phased introductions. 2 countries, Ghana and DRC, have already scaled up vaccine, and currently over 5 million children are targeted annually across the 20 countries, and we expect that to increase as more countries introduce and scale up continues. We are expecting additional introductions and scale up before the year end. 25 countries have been approved for Gavi support, including 13 for scale up. Gavi, the Vaccine Alliance, provides financial and technical support to countries as they introduce and scale up vaccines. They are right now going through a replenishment, and due to uncertainties around that replenishment, they've had to limit the support to countries for malaria vaccine scale up to 85% of the volume needed for moderate and high transmission areas. So this has resulted in the countries really having to make some difficult decisions about what areas not to introduce the vaccine in. All countries that are introducing the malaria vaccine include messaging about the importance of continuing their package of malaria interventions. Remember, the vaccine reduces malaria by about a half during the first year after introduction, with the 4th dose prolonging that protection. So it's really important to continue using bed nuts and seeking care should fever develop. So I will stop there. I want to thank all of you. These are photos of all the first children in each of the countries that introduced the vaccine. So quite a joyful slide.
I hope, Mary, you can stay to the end, because the questions will be at the end if we have some time after. But thank you very much for this excellent talk. And I think we are about to hear now more good news. And this is from Andy Pollard talking to us about typhoid and paratyphoid vaccines.
Great, thank you very much, Ron, for the introduction. I'm going to talk to you about typhoid and paratyphoid vaccines. And there is a link to Bucharest. And many of you may be aware that Ion Cantacuzino was a real important force in microbiology and experimental medicine here in Romania, who studied in Paris, and he worked under Mechnikov, who's won the Nobel Prize on understanding innate immunity. And then he returned to Bucharest, and he really pioneered a lot of work here around anti-sera for streptococcus and diphtheria, and he worked on vaccines for typhoid, cholera, diphtheria, and then the rollout of BCG here in Romania. So a really important figure, and really reminds us that in the 19th century and early 20th century, here in Europe, typhoid was a major problem all across the continent. And indeed, in the latter half of the 19th century, where I live in the UK, typhoid was an enormous burden of disease across all cities, as well as cholera. And this fellow, William Jenner, wrote a really important treatise trying to describe the clinical differences between typhoid and typhus, two very similar conditions. Having seen both of them in low-income settings myself, I really don't think I could tell the difference early in the illness. But maybe if I'd had a copy of his treatise with me, I could have worked that out. Now, when we look at the burden of typhoid around the world, one of the problems is we have very poor tools at being able to establish how many cases there are. And in the red dots up here across 3 countries, Malawi, Nepal, and Bangladesh, we've looked in 100,000 people in each country to look at the burden of typhoid in the population. And you can see across different age groups, the ages are across the x-axis at the bottom for the 3 countries. And you can see a higher rate that's in particularly school-aged children and preschool children. And those rates reach somewhere in the highest around 400 or 500 per 100,000 population. But if you then do the adjustments to look at the way people seek healthcare, whether a doctor will actually take a blood sample, and lastly, whether the culture will be positive. We know only about 50% of cultures will turn positive, even when we know there's typhoid in there. And when you put all of that together, you realize the burden is possibly as much as 10 times higher than we can measure directly with these type of burden studies. So this is an enormous burden on the population and on healthcare systems with undifferentiated fever, really difficult to distinguish from other infections in these settings. Now, we have had vaccines for typhoid for a long time. The first developed by Armworth Wright in 1896 was a heat-killed typhoid vaccine which was used in the Boer War in South Africa for troops. And that was then modified by David Harvey of the Royal Army Medical Corps in England and used from about 1915 in troops in the First World War. So there is a long history of these vaccines being used, but these were very reactogenic and people didn't tolerate them very well. And in the latter half of the 20th century, we had development of a live attenuated vaccine, TY21A, which is almost exclusively today used as a traveler vaccine. And then polysaccharide vaccines made from the sugar coat on the surface of typhoid, which is from the virulence determinant, the VI antigen on the surface, the polysaccharide. And those are also largely used for travelers. Now, because we had these vaccines available and there was an increasing recognition of a big burden of disease, back in 2008, WHO said, well, we should just start using these vaccines that we have already. It's such a big problem around the world with very large numbers of cases, maybe 11 or 12 million cases of directly measured and tens of thousands of deaths. But there was a new generation of vaccines, the conjugate vaccines, rather like the Hib or pneumococcal or meningococcal vaccines we use today for childhood immunization. And these were being developed during the 1990s. And they look very promising. So actually, people didn't want to move ahead with the old vaccines. They wanted to wait for this new vaccine. But unfortunately, the promise of the early trials didn't turn into a product. And in fact, this particular vaccine, which really led the charge, has never been developed further. It's currently it was developed at NIH in the US, and it's currently with a Chinese manufacturer who's not taken it forwards since the late 2000s. So roll forward to the last decade, and there's been some development in India of conjugate vaccines using that technology. This is the first of them from a company called Bharat Biotech, who developed a conjugate vaccine for typhoid, again, based on the polysaccharide capsule. And that vaccine was licensed in India and used a bit, but actually there wasn't huge confidence in its use, and it really needed some catalyst to get people confident in its rollout and wider use. And WHO in its guidance for developers said, well, perhaps you could improve confidence in vaccines using human challenge models. And these are models where first of all, the pathogen is grown in a laboratory under very careful conditions, so you know exactly what's in the vial before you give it to volunteers. We then ask our volunteers to come to the clinic, and we neutralize gastric acid, as you can see here. These 2 individuals drinking some sodium bicarbonate solution. And then we grow the bacteria from the vial that we've got frozen down in the laboratory and suspend it in 30 mils of bicarbonate. So this is a bit like now if you were drinking contaminated water in South Asia. And so we then ask our volunteers to drink the contaminated water, which they're doing here, and observe them over the course of the next 2 weeks. Now, of course, we do pay them for taking part. This is something which appears in the newspapers whenever we start these studies, and it helps with the recruitment. I think the newspapers are trying to say that we're being unethical in some way, but actually it's very good for recruitment of these studies. And then we monitor disease in the volunteers. We monitor them very closely. We take blood samples every day, looking for the onset of typhoid. And some volunteers will develop no infection at all. This is the temperature chart of an individual who doesn't develop any infection. You can see that the black line doesn't go above the red 38-degree line. In the box at the bottom, you can see that there's some typhoid detectable in the stool at the time of challenge. And then with these volunteers, some will develop typhoid. This is a typical typhoid case. About 7 to 10 days after ingestion of the bacteria, this individual develops a fever. The green line is a rise in CRP. And you can now see that there's more typhoid in the stool, the orange boxes, but also we can grow it from the blood of these individuals now. So this is very classical typhoid infection. And thirdly, some individuals have no symptoms at all, but they do develop blood culture positive typhoid, and we can only tell that because we're taking samples from them every day. Otherwise, they wouldn't know that they were unwell. So there's three main categories that we're seeing in the challenge model. And using this model, we can infect somewhere with 10,000 bacteria, somewhere around 2/3 of individuals will become infected. The other third presumably have some innate mechanisms, or maybe they've got a bit more gastric acid than other people, and they don't develop infection. So about 2/3 develop infection, and the other third don't. So now we have a model established, and we started work on this back in 2011, that we can get reproducibly about 2/3 infected. So we can now take a new cohort of individuals and test vaccines in the cohort. And the way we do that is we vaccinate them first of all, we wait for 28 days, and then we challenge them with typhoid, getting them to drink the bacteria, and monitor them closely, just as I described before. And what we find over the period of the next 2 weeks is that we are able to prevent infections with vaccination. The red line on this Kaplan-Meier plot are those individuals who are not vaccinated, and as before, around 2/3 of them still develop infection. But the blue line at the bottom are those individuals who are vaccinated, and we're able to demonstrate protection in the model. The model's really stringent. We're giving them a high dose of bacteria, we're monitoring them really closely, and so we're able to pick up cases more readily than you could in a field study, and yet the vaccine's still able to provide protection. Now, I haven't got time to go into a lot of detail, but there's a huge amount of work that we can do when you've got this experimental model where you can see the individual right from the before they're exposed to a pathogen through that whole period of time that they're developing developing infection, and then the immunity that develops afterwards. Or in the case of the vaccine study, to look at which bits of the vaccine immune response relate to protection. And we've shown in this model that a combination of IgG against the capsular polysaccharide and IgA is strongly correlated with protection, as is the engagement of neutrophils, which are able to phagocytose the bacteria. And this is through the Fc alpha receptor, so the IgA receptor, we think, on neutrophils. So if you have neutrophils, IgA and IgG, that seems to give you good protection. But I haven't got time to go into the data behind that. We also look at different antibiotic regimens, and we find that fever clearance and bacterial clearance is much faster with fluoroquinolones like ciprofloxacin than it is with the azithromycin. Unfortunately, today, 15 years on from the first studies, most of the typhoid in the world is resistant to ciprofloxacin. So although it performs better with a sensitive strain, it's not useful to use it in the field. So with this information showing now that we've got a vaccine that's licensed in India, it's immunogenic, and extra confidence that it does actually protect against typhoid, the World Health Organization approved its use and made recommendations that it should be used among all children under the age of 15 in a catch-up campaign, and then for routine use at around 12 months of age. And so this then policy decision started to galvanize countries to apply to Gavi for funding to start programs. And then the pandemic came, which slowed things down a bit. But in the meantime, we were saying, well, okay, we've done the challenge model, but it really would help countries to know about performance in the field. And so we started studies in around 100,000 children across the 3 countries that we'd been doing surveillance in. And you can see those 3 countries again, Nepal, Bangladesh, and Malawi, with a fantastic team of investigators working with us in each country. And we're able to then monitor the efficacy in the real world, so not in a challenge model now, but in the real world in children under the age of 15 in those low-income settings. And those studies showed that the vaccine had around 80% protective efficacy. So highly effective in preventing typhoid over a 2-year period in vaccinated children. And that really was very important in the galvanizing and catalyzing the movement towards rollout of these vaccines. The studies showed very similar results across all 3 countries. And these are the publications from the countries. But really importantly, in Bangladesh, the size of the trial was large enough that we were able to show that there was no age effect. So even in the youngest children, and you may not be able to read this, but it includes those under the age of 2, we saw that same 80% efficacy of the vaccine. So very good news for the initial rollout of the vaccines that you'd expect impact regardless of age. But one bit of disappointing news is that in the design of the trial in Bangladesh might have been able to pick up herd immune effects. Would you get extra benefit of the vaccine by protecting the unvaccinated if you reduce transmission? And we couldn't show any evidence in that trial of herd effects. But despite that, there's been a huge movement over the last 5 years. And by the end of this year, it's expected around 120 million doses of that vaccine, or in fact now there's some others on the market which have very similar properties, will be rolled out across those regions that you can see shown there on the map. Some of them have application process and haven't actually started rollout, but many millions of children, around 70 million already vaccinated today, and we should be up to 120 by the end of the year. Now, the Salmonella vaccine pipeline is quite complicated because I've talked about typhoid, which is the biggest burden, but there's also non-typhoidal Salmonella, which is a big problem in infants and toddlers in Africa, and also paratyphoid, which in school-aged children is a big problem, about 30% of the syndrome of enteric fever in South Asia. I'm just going to talk very briefly about the vaccines for paratyphoid. Now, paratyphoid is, as I said, about 30% of disease, but there is some concern that when typhoid vaccines are rolled out, that it will expose a niche which will allow paratyphoid to become more common. And we have some observations of that. This is in China, where, in fact, the old vaccine for typhoid was used on a large scale in this region. I'm not sure I have a pointer, but if I could, I would be pointing that the line that goes down is typhoid going away. The line that goes up is paratyphoid starting to emerge at the same time. And we found in some of the countries where the typhoid vaccines have been rolled out over the last 5 years that we have seen an increase in paratyphoid. It is very difficult to be sure that the two are linked because there are some countries which are not using any typhoid vaccines which have also seen a rise in paratyphoid. So I mentioned David Harvey's vaccine from 1915, and it in fact covered typhoid and paratyphoid. So it's not true to say we've never had a paratyphoid vaccine, but it's true that there is no paratyphoid vaccine today that's available or in use. And that vaccine, in some traveller studies, indicated that it may prevent up to 70% of infections. Very small numbers, and it's difficult to know whether you can believe this, but this was a heat-killed vaccine, and it appears, at least in this traveller study, to have had some impact. And we've looked at individuals who have previously been in a paratyphoid challenge model, And then when we come back and we give them a second chance to drink paratyphoid, we find that there is some protection in those individuals, about 50% protection. So that does suggest that prior exposure might protect you from further cases of paratyphoid. So I just want to very briefly mention a live attenuated paratyphoid vaccine, which is one of the different modalities that's being developed on the technologies. And this has been developed by Mike Levine and his team at the University of Maryland. So we've been collaborating with him for a number of years now, and so we brought that vaccine into the paratyphoid challenge model, very similar to the typhoid challenge model. And we found this new live attenuated vaccine, which is again taken orally, to be very safe and well tolerated after both the first and the second dose. We gave it in a 2-dose regime. And in this preliminary result, we saw around about 70% efficacy of this vaccine in the challenge model. So there's some formulation work to do, but it's very encouraging that we today have the first paratyphoid vaccine that has shown some efficacy. Field studies not done so far, but at least in the challenge model. And interestingly, it also reduced stool shedding excuse me, of paratyphoid, which might indicate that it could reduce transmission. That obviously needs further investigation in the field. The vaccine was immunogenic, and we have some work going on correlates to try to understand what part of the immune response might have been related to the protection that we saw in those individuals. So what about conjugate vaccines? So there is no capsule on the surface of paratyphoid to make a capsular conjugate vaccine, a polysaccharide conjugate vaccine, but it is decorated on the surface, like all gram-negatives, by lipopolysaccharide, or the O antigen of lipopolysaccharide. And so the several developers have been trying to make a bivalent vaccine that has as a conjugate of the typhoid that I've just talked about, but also combined in the same syringe with an O antigen conjugate. And the leading candidates of this technology are from Serum Institute of India and Biological E, another Indian company. And the clinical development plan for these vaccines is to do a phase 1 study, and then to test in the human challenge model at the same time as phase 3 safety studies, or just before, and then to go for licensure in India, followed by WHO and pre-qualification. And both of those companies I mentioned have already done their phase 1 trials, and this is the data from the phase 1 trial from Serum Institute of India. We did the immunology for this in my laboratories, so we have good information about this. And essentially, there are very strong immune responses to both the typhoid and paratyphoid components. What we don't know is whether O antigen, the paratyphoid part of this vaccine, will protect or not. And the World Health Organization has said, well, it's going to be too difficult to do a paratyphoid trial in the field to prove efficacy. So we're going to have to rely on the challenge model to decide whether to go ahead with the use of bivalent typhoid, paratyphoid conjugate vaccines. And so we have just started work on with one of these vaccines, the Serum Institute of India vaccine, on testing in the challenge model all across the UK. We have 10 sites opening across the UK to do this study. And the idea with the paratyphoid component will be licensed on the basis of efficacy in the challenge model and safety in India, whereas the typhoid component can be licensed on the basis of non-inferiority against the currently licensed typhoid vaccines. And so I think this will be very exciting, the first vaccine for global health use that might be licensed for a challenge model, if indeed it works and it protects against paratyphoid. Millions of children today are being vaccinated against typhoid as we speak, and we are at a really important moment for the paratyphoid vaccines of the first studies underway in this era that might tell us whether they work. And the phase 1 data for those vaccines is promising, at least that they're immunogenic. So it's only going to take 1 or 2 years from now before we have a good idea about these new vaccines. But really, one of the frustrating things about this whole area is in this field, clean water would solve the problem, not only for typhoid, but many other enteric pathogens. And we still feel much further away from that than we do from a typhoid or paratyphoid vaccine. Lots of people to thank, but I've run out of time, and thanks very much for inviting me to speak.
Okay, thank you very much, Andy. Just a reminder, we will have the Q&A session at the end of the table, and don't forget that you can send the questions through the app. And now the third lecture would be about the protection against GBS, Group B Streptococcus, and the speaker is Paul Heath. Thank you, Paul.
Thank you very much, Javier. Good afternoon, everyone. It's a great pleasure to be here. I note that Andy and I, and indeed Javier, are wearing the same shirt, but it was my idea first. So I'm very pleased to be talking about progress towards a Group B streptococcal vaccine. So to begin, my declarations. And so I'd like to just remind you about the pathophysiology of Group B strep infection in young infants. I think this audience, I'm sure, knows this well, but we divide Group B strep disease in the first 3 months of life into early onset and late onset disease. And early onset disease is the first 6 days of life, and late onset disease thereafter. And around 70% or so of disease in the first 3 months of life is in the first week of life. That depends a little bit on the use of intrapartum antibiotic prophylaxis in different populations. In terms of the mechanism, early onset disease reflects vertical transmission, so ingestion, aspiration, or contact in the birth canal with Group B strep. We know that 20 to 25% of women are colonized with Group B strep. Around half of the babies born to these mothers are then colonized, and around 1 to 2% of babies develop invasive disease. Colonization also predisposes to preterm births, to stillbirths, and to maternal infections. Late onset disease reflects both vertical or indeed horizontal transmission, so from the hands of healthcare workers, parents, fomites, and indeed breast milk. There is a global burden of Group B streptococcal disease. It is a significant cause of morbidity and mortality in infants, representing or presenting with sepsis, meningitis, and pneumonia. And these are some global estimates for the year 2020. So 231,000 early onset cases, 162,000 late onset cases, 46,000 stillbirths, 518,000 preterm births, 91,000 newborn deaths, which is about the same as RSV if less than 5 years of age, and then 40,000 babies with long-term disabilities as a result of meningitis or indeed sepsis due to Group B streptococcus. There is also a significant burden in high-income countries, and I'll give you 2 examples. One is the UK with a risk-based strategy for preventing early onset disease with intrapartum antibiotics, and the other will be the US with a swab-based screening approach to IAP. So in the UK setting, we have an increasing burden of infant GBS disease. It's the most frequent cause of early onset infection. It's the most frequent cause of meningitis in infants in the first 3 months of life, the most frequent cause of bacterial infection-related deaths in infants, and indeed it is an increasing cause of infection in our infants. And so 2 national surveillance studies that we did over a 15-year period demonstrating an increase over time in incidence. In the US, where the prevention is through a swab-based screening policy, there has been a significant impact of IAP on early onset disease, as you can see here in the blue line. The early onset disease, however, has persisted subsequently, and late onset disease has not been impacted by intrapartum antibiotic prophylaxis. And this just brings us up to the more recent time, and you can see that persistent burden, and it also reminds us of the burden in postpartum women and women during pregnancy. Indeed, very recent data from the US shows the persistence of Group B strep as a cause of meningitis in that population. That is the blue line there. So this is undoubtedly a burden that needs to be prevented, and we have 2 broad strategies at the moment. We have intrapartum antibiotic prophylaxis, and we have the potential of vaccination in pregnancy. And as I've shown you, intrapartum antibiotic prophylaxis is not effective against late-onset disease, stillbirths or preterm births. It carries with it an increased risk of antimicrobial resistance, as shown with erythromycin and clindamycin in particular, and then the potential adverse consequences of the impact on the infant microbiome. And intrapartum antibiotic prophylaxis is not easily achievable in low and middle income countries where the burden is considerable. And then overall, even in those countries that can administer IAP, the incidence of early onset disease and meningitis has persisted or increased despite the use of IAP. In contrast, the potential of vaccination is that it will impact on early onset disease and late onset disease, preterm births, stillbirths, and maternal disease. And we have 2 vaccine candidates with the potential to cover nearly all of the disease-causing strains, as I'll show you shortly. And indeed, with these vaccines, phase 2 trials have been completed in pregnant people, showing them to be immunogenic and safe. And the phase 3 trials are awaiting regulatory feedback, and I will certainly return to that. But to go back to the start, so the possible targets for a Group B strep vaccine are these. This is an encapsulated bacteria. There are 10 serotypes, and then there are also conserved surface proteins, and both of these serve as potential targets for vaccines. And indeed, the history, the rationale goes back 90 years or so when Rebecca Lansfield first demonstrated that infection in mice could be, protection against infection in mice could be achieved using capsular polysaccharide-specific rabbit sera. And then in 1976, Carol Baker showed that low levels of naturally occurring maternal antibody to the capsular polysaccharide correlated with susceptibility of the neonate to GBS disease. So we must really acknowledge the importance of Carol Baker's work over this time in demonstrating this relationship. And of course, this reflects placentally transferred GBS IgG antibodies. Indeed, Carol and her colleagues did a huge amount of work in the 70s, 80s, and 90s, developing monovalent and then bivalent and trivalent conjugate vaccines against the important serotypes, including giving such a vaccine to pregnant women. That then was taken forward by Novartis and GSK for a time using the CRM197 conjugate vaccines. This is the first pregnancy study that was done by Carol Baker and her team, and this is with a serotype 3 tetanus toxoid conjugate vaccine. And what you can see here is the geometric mean concentration, GMC, antibody in pregnant women and then in infants after birth. So before vaccination at 30 to 32 weeks, very little antibody, and then antibody at delivery in the cord blood of the infant, and then out to 2 months of age in babies born to vaccinated women. There is a placebo control group there, but you can't see the antibody. So this showed for the first time the potential for such a vaccine to protect infants out to at least 2 months of age, which of course covers the bulk of the late onset Group B strep period. It does also beg the question, of course, as to how much antibody is required to protect infants. And I will return to this, a serocorrelative protection. But here, using a fairly conservative estimate of 0.5 microgram per mil, you can see that these levels of antibody are well above that. So this is the current status of GBS vaccine development. And you can see the 2 broad platforms, the conjugate vaccines and the protein subunit vaccines. So in terms of the conjugate vaccines, there are some vaccines in preclinical development and Invent-Price in phase 1 clinical trial. But the most important player is the Pfizer hexavalent vaccine, which is now poised to go into phase 3 trials. And then in the protein subunit vaccine platform, we have the Minivax vaccine, and that too has now gone through phase 2 and is poised to move into phase 3. And also on the slide, you can see the other important milestone. So the recognition by the World Health Organization of the importance of Group B strep globally, the preferred product characteristics, the roadmap for implementation of Group B strep vaccines, and then the recognition by regulators of the importance of these vaccines. So this is a summary of the GBS6 hexavalent capsular polysaccharide conjugate vaccine, which is the Pfizer vaccine. And this consists of the capsular polysaccharide from 6 important serotypes, individually conjugated to cross-reactive material 197, the mutant diphtheria conjugate protein. 20 micrograms per serotype. The proposed schedule is 1 dose in pregnancy and no adjuvant is required. And as I mentioned before, it is estimated that coverage of this vaccine would be very high across all the disease-causing strains. So it also shows the preclinical phase with the demonstration in an animal model of the efficacy of this vaccine, the demonstration that there are no developmental or reproductive toxicity concerns in animal models, and then the phase 1 and 2 trials, including trials now in women living with HIV. And then we await the phase 3 trial to the right there. And then when it comes to the protein vaccine, this is the Minavax vaccine, and it is based on a family of 6 surface proteins, the alpha-like proteins, the ALP proteins. The vaccine itself utilizes the N-terminals of 4 of these ALP proteins combined into 2 fusion proteins, so GBS-NN2. The proposed schedule is 1 or 2 doses during pregnancy, and this does have an adjuvant, an aluminum hydroxide adjuvant. And again, the expected coverage of disease-causing strains is very high. There have been 2 parts to this story, with the initial development of a 2-ALP protein vaccine, but then the recognition that it needed to cover 4 ALP proteins, which is at the lower end of the bottom of the slide here. And then the demonstration in animal models of the efficacy of this vaccine, the lack of developmental and reproductive toxicity concern, phase 1 and 2 trials, again, including trials now in women living with HIV, and also now poised to enter phase 3. So the issue of licensing this or these vaccines. So we have 2 vaccines which look like excellent candidates, showing immunogenicity and safety in the studies that have been demonstrated so far, showing placental transfer and persistence in the infant born to vaccinated women. So what is the issue here? The issue in terms of licensing this vaccine is that a traditional phase 3 efficacy trial with a clinical endpoint is probably too difficult to do, because although this is the most common cause of early onset infection and of meningitis in infants, the disease incidence is relatively low, and so a suitably powered vaccine efficacy trial is indeed very high. And there are some numbers here to demonstrate that. So even with a population disease incidence of 2 per 1,000 live births, which is a sort of incidence that South Africa did have before, and thus very high, and making various assumptions about coverage of the serotypes and the efficacy of the vaccine, it would require 40 to 60,000 pregnant women in such a trial. And of course, we need to recognize the stringency of the trial and the infrastructure required to do this. Very demanding. Group B strep disease happens very quickly and may happen very quickly after delivery. Important need to investigate deaths and stillbirths diligently, and of course the adequacy of safety oversight and monitoring is important. So in a population with a likely incidence of 0.5 to 1 per 1,000 live births, it would require up to 100,000 pregnant women, and that is likely to be commercially unviable. So what is the approach then to licensure? The preferred approach is based on antibody correlates of protection. And on this slide, these are data from South Africa. And what perhaps you can see is the antibody levels in cases and in controls. The controls are infants born to women who are colonized with the same serotype, but whose babies don't go on and develop disease in the first 3 months of life. And I think you can see a clear separation here for two serotypes, serotype 1a and serotype 3, with a much lower level of antibody, of course, in those that become cases of disease. And indeed, in that and subsequent analyses, on the right there, you can see the proportion of infants in the study in South Africa achieving the proposed threshold against these individual serotypes, all of which in the main are very high, suggesting that these levels, this protection, would be achieved as a result of this vaccine. Similarly, with the protein vaccine, the ALP protein vaccine, it is possible to demonstrate through natural immunity studies that there are antibody concentrations associated with protection, serological thresholds of risk reduction. So again, an approach based on defining such thresholds should be possible for licensure. And importantly, this approach is also agreed in principle by the regulators, and you can see here the FDA accelerated approval pathway for vaccines such as Group B strep, where there's an unmet medical need, a serious condition, meaningful advantage over existing therapies, which would be IAP, demonstrating an effect on a surrogate endpoint that is reasonably likely to predict clinical benefit, and then the need for post-marketing confirmatory trials to verify the effectiveness and safety. So this is the approach that is expected now. This is demonstrated in a different way. So option 3 is the traditional disease endpoint clinical phase 3 trial, but it's option 1 that is the approach that we think is likely, with an immunological endpoint trial, perhaps 6,000 or 7,000 pregnant women, which is achievable, demonstrating safety in a significant number, but also with an immunological endpoint, and then moving into an effectiveness study, a phase 4 trial in one or more model countries in which the safety and effectiveness are demonstrated. And so let me remind you again about the potential global impact of an effective Group B strep vaccine, and these are based on modeling data from Anna Seeland and others. Maternal vaccination could prevent 231,000 infant and maternal GBS cases every year, 41,000 stillbirths, 66,000 infant deaths. And indeed, in many countries and regions which have examined the cost-effectiveness of this approach, it is clear that a maternal vaccine would be more cost-effective than the currently employed prevention strategies, depending, of course, on the price of the vaccine. So just to finish, there has been considerable progress, and we're poised to move into the definitive studies and trials to make this possible now. And maternal vaccination, of course, has the potential to protect the mother, the fetus, and the infant from birth against Group B strep. There has been, of course, steady global progress in the acceptability of maternal vaccination programs more generally, and there is a very clear rationale and considerable enthusiasm, I hope, from this audience, from the PID family, and from the obstetric and others communities for the introduction of a Group B strep vaccine. And as I've told you, the most efficient pathway to achieve this is a vaccine approval based on agreed serological thresholds of risk reduction, followed by phase 4 implementation studies to demonstrate effectiveness and safety. And I would conclude by saying preparations for this are well underway. Thank you.
Thanks, Paul. I think we're hearing a lot of good news today, and that is really great. Maybe another good news now in the following talk: efficacy and safety of clesrovimab. I hope I said it appropriately. Yes. An investigational RS-virus antibody in healthy preterm and full-term infants, subgroup analysis of a phase 2b/3 trial, and this will be delivered by Andrea Guerra.
Thank you very much. Good afternoon, everybody. Today we will be presenting efficacy and safety of clesrovimab, an investigational RSV antibody in healthy preterm and full-term infants subgroup analysis of a phase 2b3 trial called CLEVER. I am an employee of MSD UK Limited, and this is my disclosure. So as you all very well know, RSV is a leading cause of serious respiratory infection and hospitalization in infants worldwide, particularly in the first few months of life. And clesrovimab is an RSV neutralizing monoclonal antibody, and this slide highlights a few of this monoclonal's important characteristics. It binds with high affinity to site 4 of the RSVF protein. It has 3 engineered substitutions in the constant region, termed YTE, that result in increased recycling of the antibody and an extended half-life in the blood. It achieves high nasal tissue distribution and concentration. And a phase 2b3 trial called CLEVER in healthy pre- and full-term infants showed that clesrovimab was well tolerated with a safety profile similar to placebo, providing protection against mild, moderate, and severe RSV disease. As mentioned before, today I will be presenting efficacy and safety subgroup analysis from CLEVER. And CLEVER is clesrovimab pivotal phase 2b3 double-blind randomized placebo-controlled study in healthy pre- and full-term infants of at least 29 weeks gestational age and entering their first RSV season. In the study, approximately 3,600 infants were randomized 2-to-1 to receive either 105 milligram intramuscularly of clesrovimab or placebo. And they were stratified by gestational age, hemisphere, and chronological age. All participants were followed up for 365 days post-dose, with the first 1,615 enrolled followed up for 515 days post-dose. Just as a reminder, the primary endpoint of the study was safety, but in addition, the study was designed to test 2 hypotheses. The first one concerning the primary efficacy endpoint of RSV-associated medical attended lower respiratory infection, or MARI, through 150 days post-dose, and the second concerning the secondary endpoint of RSV-associated hospitalization through 150 days post-dose. And these endpoints, as I just mentioned, were complemented by a range of outcomes ranging from mild to severe RSV-associated disease. But the objective of the analysis that I am presenting today was to evaluate the efficacy of clesrovimab through 150 days post-dose and the safety of clesrovimab through day 365 post-dose in CLEVER for the following subgroups: body weight and randomization, gestational age, chronological age and randomization. In this slide, you can see that baseline characteristics were balanced between intervention groups with respect to sex, age, body weight, and gestational age. 2,411 participants received clesrovimab and 1,203 placebo. Most infants were less than 6 months of age and 5 kilos or more at randomization, and most infants were 35 weeks or more of gestational age. Please try to keep in mind these figures as we move along the efficacy and safety results in the coming slides. So let's actually move to efficacy, starting by efficacy by body weight. The number of participants by subgroup is displayed in the top row of the table. And the observed efficacy of clesrovimab relative to placebo was generally comparable across the subgroup analyzed for all the endpoints, as highlighted by the overlapping 95% confidence interval. And the observed efficacy was generally consistent with the one of the overall population. Of note, the efficacy increased with increasing severity of RSV-associated disease, and again, this was consistent with the overall population. Moving to efficacy by gestational age, again, the observed efficacy of clesrovimab relative to placebo was generally comparable across the subgroup analyzed for all the endpoints, again, as highlighted by the 95% confidence interval that are overlapping, and consistent with the overall population. And we can see again that the efficacy for the more severe RSV-associated endpoints remain high across all the subgroups. For chronological age, the observed efficacy of clesrovimab relative to placebo was overall generally comparable across the subgroup analyzed, again as highlighted by the overlapping confidence interval for all the endpoints, and consistent with the overall population. The efficacy for the more severe RSV-associated disease endpoints remained high in infants with age less than 6 months. In the group of participants with chronological age of 6 months or more, the burden of disease is expected to decrease as the severity of the disease increases. And this expectation is substantiated by the declining number of cases reported in the placebo group for the most severe endpoints, as you can see displayed at the top of the table. When evaluating acute respiratory tract infection, that is the least severe endpoint and that is displayed at the bottom of the table, and where the burden of disease is anticipated to be comparable across all age subgroups, then the efficacy is consistent across the age subgroups, including the one above 6 months of age. Turning to safety results by body weight, the safety profile of clesrovimab is generally comparable between subgroups, and the proportion of participants with adverse events, including all adverse events, drug-related adverse events, serious adverse events, drug-related serious adverse events, and serious adverse event resulting in death, was generally comparable to placebo within each subgroup. As a reminder, all events of death reported in the study were considered not related to study intervention by the investigators. Turning to safety by gestational age, the safety profile of clesrovimab again is generally comparable between subgroups, and the proportion of participants with the different categories of adverse events were generally comparable with placebo within each subgroup, as you can see from the table. And finally, turning to safety results by chronological age, again, the safety profile of clesrovimab is generally comparable between subgroups, and the proportion of participants with the different categories of adverse events were again generally comparable with placebo within each subgroup. So in conclusion, our analysis showed that for each RSV-associated endpoint, the observed efficacy of clesrovimab relative to placebo was generally comparable across the body weight, gestational age, and chronological age subgroups, as highlighted by the overlapping 95% confidence intervals. Within each subgroup level, efficacy generally increased with increasing disease severity and was consistent with the overall population. Within each subgroup level, the proportion of participants with adverse events were generally comparable between intervention groups, and safety results were generally comparable across the subgroups and were consistent with the overall population. So a single dose of 105 milligrams of clesrovimab given prior to the start or during the RSV season is well tolerated and efficacious in all healthy infants, independent of weight, gestational age, or chronological age. Now, I would like to conclude by expressing our deepest gratitude to our outstanding investigators and site personnel from 192 sites across 22 countries in the globe, and most importantly, to our precious children in the study and their families. Thank you very much.
Thank you very much, Andrea. And now let's move to the last vaccine, which is chikungunya. And it's, Vera Burger is going to tell us about the final phase 3 safety and immunogenicity 12-month data of the VLA 1553, a single-dose live attenuated vaccine in adolescents in Brazil, a pivotal trial. Thank you very much.
I don't see the slides yet, so.
Can we have the slides of the next speaker, please? Technical people, please.
Oh, no? Oh, here we go. Okay. All right. Just see.
Did you try to advance?
Okay. So, thank you for the invitation of presenting our data today. My name is Vera Bürger from Valneva, and this is my disclaimer. And I'm going to present to you today our phase 3 results of our chikungunya trial regarding the 12-month data of VLA 1553, which is a single-dose live attenuated vaccine in adolescents in Brazil. This is a pivotal trial. So this trial included 754 adolescents in Brazil, and it is our first trial that was run in a chikungunya endemic area, or in chikungunya endemic areas of Brazil, and there were 18% baseline seropositive individuals included in the trial. The primary endpoint was 28 days after vaccination, the seroresponse rate, and the immunogenicity subset was followed up to 12 months in the trial. This trial is intended to support the label extension of IXCHIQ to the adolescent population based on its 6 months follow-up data. EMA already approved the adolescent indication, and FDA review is ongoing, and further regulatory submissions and reviews might follow. This is a placebo-controlled trial, and the individuals were randomized 2-to-1 into verum or placebo control. The demographic profile, as you can see, was well balanced. There were 54% of female individuals recruited. The majority were of white or other multiracial race, and the serostatus, as I mentioned before, at baseline was 18% seropositive and 82% seronegative individuals. Now turning to the safety results. The adverse events were solicited for 10 days after vaccination. The local adverse events are displayed here, and they occurred in 32% of participants following vaccination with VLA 1553. Luckily, the majority of these AEs were mild to moderate and resolved within just 2 days. The most common ones were tenderness and pain. And also important to mention is that the frequency was comparable between seropositive or seronegative participants. For the systemic adverse events, these were also in majority of mild or moderate appearance, and they resolved within 3 days. Any solicited systemic adverse events were reported by 64% of participants in the VLA arm and 42% in the placebo arm. Represented by the blue color, the majority of these were mild. And also here, important to note that solicited systemic AEs were less frequently reported in seropositive adolescents after VLA 1553 vaccination. Looking at a key symptom, the solicited arthralgia events are presented here. And we can see that the majority of these events resolved within 5 days and was of mild intensity. All episodes of the documented solicited arthralgia events resolved within 13 days. There was one event, though, of mild arthralgia that lasted 32 days. Here is a summary of adverse events, so a combination of solicited and unsolicited adverse events. Most of them were mild or moderate, so there were about 6% severe unsolicited AEs reported in the seronegative VLA 1553 participants. There were no adverse events leading to study withdrawal. There were no deaths. and looking at the adverse events of special interest, which is a cluster of symptoms reflecting chikungunya-like disease. These were reported in 11% of seronegative participants in the verum arm, and compared to 8% in the placebo arm. Generally, there was a lower frequency of severe unsolicited AEs in the baseline seronegative stratum. Also, I would like to mention that there were no vaccine-induced viremia that occurred in adolescents who were seropositive at baseline, with viremia clearing between day 8 and day 29 in seronegative VLA 1553 recipients. Now turning to the immunogenicity results. So the primary endpoint at day 29 showed that this seroresponse threshold was met, and 98.8% of individuals showed seroresponse. And this was continued, and there was a persistent immune response available for at least 1 year after vaccination in this adolescent population. So at day 365, there are still 98% of seroresponders in this trial. On the next slide, I can guide you through the geometric mean titers. So depicted in light blue are the seronegative participants in the trial, and in dark blue, the seropositive ones. And what I would like to point out is that in seronegative individuals at day 29, the titers are similar to those that seropositive individuals have at baseline, meaning that the VLA 1553 vaccination induces titer levels similar to those after wild-type infection. Moving on to the conclusions, that the seroresponse rate was persistently high in this adolescent trial up to 12 months after the single vaccination of VLA 1553. Therefore, the trial endpoint, primary endpoint, was met with 98.8% of baseline seronegative participants developing seroresponse 28 days after the vaccination. This single-dose vaccination induced a robust immune response in adolescents up to 1 year. And in seronegative individuals, the vaccination induced at 28 days after vaccination, similar to the ones observed in baseline seropositive adolescents. And with this trial, the month 12 data confirmed that VLA 1553 was generally safe and well tolerated in adolescents, irrespective of previous chikungunya virus infection. I would like to acknowledge all study participants, trial sites involved in this trial, and also very important, the primary sponsor, or principal sponsor of this trial, Instituto Butantan in Brazil, and CEPI for funding this to allow also the development of vaccine in endemic countries and low middle income countries. And with that, thank you, and back to you.
Thank you. We are a little bit short of time, but we have about 10 minutes for question and answers. So if people want to go to the microphone. Yeah, go ahead.
This is Steve Black from the Global Vaccine Data Network. I had a question and request for comment, actually, from the last speaker, is that your vaccine has been associated in adults with encephalopathy that's been associated with viremia, with virus being detected in the CSF as well as antibody in the CSF. And some of these cases have been severe. And I'd like to ask you what the company is planning to do to further evaluate that both in adults and potentially in children as the vaccine comes into wider use.
Which one we want to ask Ahmed?
Yeah, thank you for this important comment. So Valneva definitely is dedicated to thoroughly evaluate those cases and reports. FDA, EMA, and various regulators are currently evaluating whether there is a causal relationship to the vaccination in elderly with comorbidities.
The vaccine virus was in the spinal fluid. I think that's a pretty good association for causality.
That's a fact, yes, but nevertheless, the overall comprehensive evaluations are ongoing, and the company's evaluation of the risk-benefit profile remains positive for the adults, and for elderly, a decision will be taken together with regulators on how to proceed in the most safe way.
So I think we have to go one and one.
Yeah, there's one question from for Dr. Hamal. What is the postulated mechanism for increased efficacy with seasonal timing of vaccination? And if this is understood, how can this be used to design more effective malaria vaccines? We cannot hear you.
You're muted. You are muted, Marie.
No, we cannot hear you. Is there any technical problems from here? No.
Oh, sorry.
Sorry, we cannot hear you. Let's try to sort this out. Meanwhile, is there any other question?
Yeah, a question and a comment to Andy Pollard. First of all, thank you for the excellent talk. And I am very pleased to hear about the good vaccine efficacy of the live attenuated paratyphi strain, CVD strain, Mike Levine strain in your model. The question is if there is any concern of the future performance of this live attenuated vaccine in a target population in low-income countries. And what are the plans to make sure that this vaccine will be, oral vaccine, live attenuated, will be enough immunogenic and also efficacious in such a population, target population, immunogenicity studies, and even a local challenge model in such a country to make sure that these obstacles with the intestinal barrier of children in these populations can be overcome.
Thanks very much. That's a very good question. I guess there's two components to that. The first is challenge models in low-income settings, and we are actually working through that in Pakistan at the moment. Whether that will get through the final regulatory steps, I don't know, but that, at least in adults, would address that. But I think, as you've highlighted, even with an adult study with a vaccine in a low-income setting, you don't know that you're dealing with the same environment as in the child's gut. And so I think one of the challenges with the live attenuated vaccine is we may still need to have the phase 3 trial in children in order to get licensure. I think what the challenge model does for the live attenuated is it gives confidence that that approach might work. It's sort of a gatekeeper to moving forwards. The trials for phase 3 efficacy in children are somewhere between 70 and over 100,000. Some people have said 200,000. So it's not impossible with a live attenuated vaccine, but it's very challenging to get funded, particularly for a burden of disease, which is only 30% of enteric fever. So I think that's the difficulty with directly addressing that with efficacy. But maybe there are smaller studies that could be done to try and address some of the concerns that you've just raised. But I think you're absolutely right. For us at the moment, at least for immunogenicity, being in a naive population in the UK is probably closer to a naive young child in a low-income setting than an adult in a low-income setting who have lots of immunity from past exposure. But I agree completely with the comment.
Marie, did you gain back your voice? I think we hear you now.
You can't hear me.
Yeah, we hear you now. We hear you now.
Okay.
Do you?
All right, good.
Okay, so did you hear the question that you had before?
I heard. Thank you for that question. So there are some theories about why there's higher efficacy when the vaccine is provided seasonal rather than provided in the perennial transmission areas when it's provided in an age-based way. And one is, as I mentioned, just the timing, just that we know that in the 6 months after vaccination with the first 3 doses, you have high efficacy. With the RTSS, it was about 70% during that period. With R21, it was a little higher during that period. So it may just be a matter of timing. But the mathematical modelers say that they see that it's something else, that they can't recover that level of efficacy just based on timing. So a second theory is that there's something about vaccinating when a child has parasitemia. And in areas of moderate or high perennial transmission, frequently 40% or so of children will be parasitemic at any time period. And so you have a lower efficacy potentially because you're vaccinating when children already have parasitemia. When you are vaccinating in highly seasonal areas, you're vaccinating in the low transmission season. So it's unlikely that children have parasites at that time. And then the third is that there's some synergy between the vaccine, which is given before the high transmission season and prevents infection in that pre-erythrocytic stage, and seasonal malaria chemoprevention, which is given in highly seasonal areas during the high season and mops up any infections that get through. That type of potential synergy was seen with RTSS, but when they looked for this in the trial that looked at R21, they didn't see additional benefit with SMC. So the question was, how can this be used for improved efficacy of these or future vaccines? And one thing that has been tried, which relates to the question of lower efficacy if you have parasitemia at the time of vaccination, is giving a treatment dose of an anti-malarial drug that clears that blood stage infection before vaccination, so a couple weeks before. And this was tried with a different sporozoite vaccine called PFSPZ, and did boost the efficacy both in a study of adults, but also in a study of women hoping to get pregnant and subsequently getting pregnant. And then the second thing to learn from it is that perhaps innovative timing of vaccination is worth thinking about, although then you need some understanding about how best to implement.
I guess the last question would be to Dr. Heath. Okay, so we'll have Dr. Heath, and then short question there, and then we stop.
Okay, we have plenty of questions for Dr. Heath. I am trying to mix them all. Phase 3 trials need to demonstrate antibody concentrations in both mothers and infants, and together with this, the serological assays for the 2 advanced vaccines are distinct. The antigens being so different, so there will be no possibility to demonstrate non-inferiority. Is there any possibility that commercially one takes over the other?
Thank you. So the first question was about antibody concentrations in infants and mothers, and we recognise that the correlate should be the antibody level in the infant, so cord blood typically. So that would be what would be assessed in such phase 3 trials. But I would imagine in a subset also maternal antibody, because placental transfer, which is of course variable, should also be assessed. But the basis of it is infant antibody. For the second question, yes, I think it's wonderful to have 2 or more vaccine candidates heading towards phase 3 and then hopefully implementation. Timing is of the essence, and there is of course a battle between the two with regard to that. The licensure I would envisage would be different, because the assays, as the questioner has pointed out, will be different between the 2 vaccine companies. So I guess it's potentially possible that the phase 3 and phase 4 trials will be done in different places, and then the data that emerges in terms of effectiveness, safety, the schedule, which may be different, and of course the cost effectiveness, which may also be different too, will perhaps allow different countries to choose which ones they employ. Thanks.
Okay, and very short question, please.
Thank you for the encouraging session. A question to Andy. There are advantages and disadvantages of an oral typhoid vaccine versus a parenteral VI conjugate vaccine. Any idea of doing comparable head-to-head studies to see which is better for developed countries or for developing countries?
There are no plans for those sorts of studies at the moment, just because of the cost to do that. I think you're right. It would be interesting to see head-to-head studies. I think if these different products are all licensed, then it'll have to be post-licensure studies, but difficult to do in the settings where there's a lot of typhoid.
Thank you.
Okay, thank you very much then. Thank you very much for coming, and see you in the next session.